How biological diversity evolves. Why does evolution matter? The classification system used by biologists shows how all forms of life on Earth are related. Natural selection is known as a microevolutionary mechanism. Natural selection explains the striking ways in which organisms are suited to their environment. There have been several advances since the publication of Darwin's On the Origin of Species by means of natural selection. In the field of molecular biology, scientists have given us a wealth of new information about the evolution of life on Earth. Researchers have explained the genetic patterns underlying the homology of vertebrate limbs. Hundreds of thousands more fossils have been discovered and cataloged. Researchers have been able to investigate the genetic material of certain fossils, and new dating methods have shown us the age of fossils. What is a species? Speciation is the process in which one species splits into two or more species. Species is a Latin word meaning kind or appearance. Although the basic idea of species as distinct life forms seems intuitive, devising a more formal definition is not so easy. A species is a group of populations whose members have the potential to interbreed with one another and produce fertile offspring. For example, chimpanzees and humans, despite having shared evolutionary history, are distinct species because they cannot successfully interbreed. We cannot apply the biological species concept to all situations, however. It excludes organisms that only reproduce asexually, and fossils are obviously not currently reproducing sexually, so they cannot be evaluated by the biological species concept. In response to such challenges, biologists have developed other ways to define species. Species can be classified based on measurable physical traits, such as number and type of teeth or flower structures. Another approach defines a species as the smallest group of individuals sharing a common ancestor and forming one branch on the tree of life. Yet another approach proposes defining species solely on the basis of molecular data, a sort of barcode that identifies each species. Reproductive barriers exist between species. A reproductive barrier is anything that prevents individuals of closely related species from interbreeding. We can classify reproductive barriers as either prezygotic or postzygotic. Prezygotic barriers blocks interbreeding before the formation of the zygote or fertilized egg. Postzygotic barriers blocks interbreeding after the formation of the zygote or fertilized egg. Prezygotic barriers prevent mating or fertilization between species. This includes temporal isolation, habitat isolation, behavioral isolation, mechanical isolation, and gamete isolation. Time-based or temporal isolation means that different species typically breed in the wild at different times. Habitat isolation prevents breeding because different species do not live in the same area. Behavioral isolation involves the differing reproductive behaviors. Different mating rituals and customs are seen in different species. There is also mechanical isolation or physical incompatibility. The act of mating may be physically impossible or improbable between different species. And there's fertilization prevention or gamete isolation. The gametes, which are eggs and sperm, are different in different species and are often incompatible. Postzygotic barriers operate if interspecies mating actually does occur and results in a hybrid zygote or fertilized egg. In these cases, a hybrid is formed. A hybrid means that the egg comes from one species and the sperm from another species. Often we see the post-zygotic barrier of reduced hybrid viability, meaning that some hybrid offspring die before reaching reproductive maturity. The offspring fails to develop normally because of the genetic incompatibilities between two different species. In other cases of hybridization, offspring may become vigorous individuals, but are infertile. A mule, for example, is the hybrid offspring of a female horse and a male donkey. Mules are sterile. They cannot successfully breed with one another. Another post-zygotic barrier is reproductive isolation. A population can sometimes get cut off from other populations of a parent species. With its gene pool then isolated, the splinter population can follow its own evolutionary course. Reproductive isolation results from two general scenarios: allopatric speciation and sympatric speciation. Allopatric means different country, and sympatric means same country. In allopatric speciation, the initial block to gene flow is a geographic barrier that physically isolates the splinter population. In sympatric speciation, the origin of the new species is without geographic isolation. The splinter population becomes reproductively isolated even though it is in the midst of the parent population. In allopatric speciation, a variety of geological processes can isolate populations. For example, the water level in a large lake may subside until there are several smaller lakes, each with a separate population of fish. A stream may change course and divide populations of animals that are unable to cross the water. Over time, a river flowing over rock may carve a deep canyon that separates the inhabitants on either side. Continents themselves can split and move apart. And the rise and fall of sea levels can submerge or expose land bridges between continents. Speciation is more common for small isolated populations. For each small isolated population that becomes a new species, many more simply perish in their new environment. The population may adapt to its local environment and begin to look very different from the ancestral population, but that doesn't necessarily make it a new species. Speciation occurs with the evolution of reproductive barriers between the isolated population and its parent population. In sympatric speciation, a new species arises within the same geographic area as its parent species. Factors that can reduce gene flow in sympatric populations include polyploidy, habitat complexity, and sexual selection. In polyploidy, a species originates from an accident during cell division that results in an extra set of chromosomes. New species formed in this way are considered polyploid, meaning their cells have more than two complete sets of chromosomes. Some animal species, especially fish and amphibians, are polyploidy. In fact, 80% of present-day plant species are descended from ancestors that arose by polyploid speciation. Two distinct forms of polyploid speciation have been observed. In one form, polyploidy arises from a single parent species. A second form of polyploid speciation can occur when two different species interbreed and produce hybrid offspring. Polyploid speciation can occur when two different species interbreed and produce hybrid offspring. Most instances of polyploid speciation in plants resulted from such hybridizations. A mule is sterile because its parents' chromosomes don't match. A horse has 64 chromosomes. A donkey has 62 chromosomes. Therefore, each mule has 63 chromosomes. These structural differences between the chromosomes of horses and donkeys prevent them from pairing correctly. and the odd number leaves one chromosome without a potential partner. Consequently, mules don't produce viable gametes. However, we don't see the same issue in sympatric speciation in plants. A hybrid's chromosome in plants are not homologous and cannot reproduce sexually. However, the hybrid may be able to reproduce asexually, as many plants do. Many of the plant species we grow for food are polyploids, including oats, potatoes, bananas, strawberries, peanuts, apples, sugarcane, and wheat. The wheat used for bread is a hybrid of 3 different parent species and has 6 sets of chromosomes, 2 sets from each parent. Plant geneticists use chemicals to induce errors in cell division and generate new polyploids in the laboratory. By harnessing this evolutionary process, they can produce new hybrid species that have desirable qualities. Volcanic islands such as the Galapagos and Hawaiian island chains initially had no life forms. Over time, colonists arrived via ocean currents or winds. Some of these organisms gain a new foothold and establish new populations. In their new environment, these populations may diverge significantly from their distant parent populations. In addition, islands that have physically diverse habitats and that are far enough apart to permit populations to evolve in isolation, but close enough to allow occasional dispersals to occur, are often the sites of multiple speciation events. The Galapagos Islands were formed by underwater volcanoes from 5 million to 1 million years ago. These islands are one of the world's great showcases of speciation. They are home to numerous plants, snails, reptiles, and birds that are found nowhere else on Earth. The Galapagos Islands have 14 species of closely related finches. These birds share many finch-like traits, but they differ in their feeding habits and their beaks, which are specialized for what they eat. For example, the woodpecker finch uses cactus spines or twigs as tools to pry insects from trees. And the thin, probing bill of the warbler finch is well suited for capturing small insects. These finches differ in their habitats as well as their beaks. Some live in trees, and others spend most of their time on the ground. Once these finches were completely isolated on the island, the founder population may have changed significantly as natural selection adapted it to the new environment, and thus it became a new species. Later, a few individuals of the new species may have migrated to neighboring islands, where, under different environmental conditions, a new founder population was changed enough through natural selection to become yet another species. Some of these birds may have recolonized the first island and coexisted there with the original ancestral species, if reproductive barriers kept the species distinct. Multiple rounds of colonization and speciation on many of the separate islands of the Galapagos probably followed. Today, each of the Galapagos Islands has several species of finches, with as many as 10 on the same island. Reproductive isolation, due to species-specific mating songs, helps to keep the species separate from one another. Speciation is a very slow process. In contrast, microevolutionary changes can be apparent in a population with only a few generations. The species living today represent only a brief instant in the vast span of time. The environment continues to change, sometimes rapidly due to human impact, and natural selection continues to affect populations. It is reasonable to assume that some populations are changing in ways that could eventually lead to speciation. Studying populations as they diverge gives biologists a window on the process of speciation. Researchers have documented at least 24 cases in which populations are currently diverging as they use different food sources or breed in different habitats. Numerous cases involve insects exploiting different food plants. In one well-studied example, a subpopulation of a fly that feeds on hawthorn fruits found a new resource when American colonists planted apple trees. Although the two fly populations are still regarded as subspecies, researchers have identified mechanisms that severely restrict gene flow between the two. In other cases, biologists have identified animal populations that are diverging as a result of differences in male courtship behavior. In one survey of 84 groups of plants and animals, the time for speciation ranged from between 4,000 to 40 million years. Speciation may begin with small differences. As speciation occurs again and again, these differences accumulate. This accumulation may eventually lead to new groups that differ greatly from their ancestors. Thank you for watching.